Hi, everyone. This is Rupert Koopman on behalf of the Botanical Society of South Africa for our first BotSoc webinar on the year. I'm just checking with my people that there was a small glitch in terms of sound. Sounds okay. If you can hear me, let me know. Welcome to 2023. And what a time. Oops. And that was YouTube catching up with us. So everything seems a go, and I think the latency is 23 seconds, so 2023 again. So what we're up to this year is to immediately teach you a new word, because I think, like all of us, herbariums was what we thought was the plural, but it's actually herbaria. So thank you to all the herbaria workers who pointed that out to me. And I'm very thrilled to hear the kind of back-to-back or back behind the scenes operations of the herbariums, because I think, unlike I had the privilege of interning at the Kirstenbosch Research Center and spending a lot of time behind the scenes at the Compton Herbarium. Um, Most of us don't really think about what happens after the plants are pressed and have been set in. So I want to thank in advance today's presenters for lifting the lid on that. Due to load shedding, we've pre-recorded all the presentations, so I will be doing that. And if people could also just check if you could if you can send messages in the chat. If so, please say where you're from. Our housekeeping rules are pretty simple. We're going to be playing these three presentations back to back, and then please put any questions that you have in the Q&A, and then we'll see how we go from there. So right now, we're going to go start with one of the oldest herbaria in the country. And definitely one of the largest, the Compton Herbarium. And let's fix the chat quickly. Okay, I will fix the chat as soon as we start the presentation. Okay, try the chat now, everybody. Let's go. And then let me go to the first presentation, which is Henri Marais, who's, Marais Tienes. I know her as Marais, which shows how old I am, who's a herbarium technician at Compton. And let's see what's happening down here at Kirstenbosch. Okay, take it away, Henri. Your signal.
I think it's my internet. I'm having issues. It kicked me out of Zoom for a bit. It's currently load shedding, so can you hear me now?
Yeah,
Hello,
not
can you hear
well.
me?
I think what we're gonna do is I'm gonna go into Jenny's one, and then you and I see what we can do behind the scenes to sort things out.
Okay.
Okay, good.
Okay.
Thank you.
Sure. Thanks.
Okay, everyone, apologies for that. Let's try and get to the next speaker. That's Jenny Potgieter from Southern Cape Herbarium. And we'll see what we can do to restore this.
Good afternoon. I'm Jenny Potgieter from George. I've been involved with the Southern Cape Herbarium as a volunteer from 1996. I'm not a botanist, but have a love of plants, especially the fynbos, and presently I run the Outram's crew group in George. In 1994, Yvette van Wyk, a local botanist, who had recently moved to the area, discovered that the herbarium collection at Sarsveld College had been moved to Pretoria. With her insistence and the help of Jan Vlok, the collection was returned to George. The Southern Cape Herbarium was then started with these specimens, most of which were trees. Initially, it was housed in the George Museum in what we call the cupboard, a dark and dingy place. It was totally unsuitable, but all that was available at the time as we were unfunded. A few years later, Audrey Moriarty, the author of The Wildflower Guide of the Southern Cape, and a friend of Yvette's, left enough money to allow for the purchase of a house to be used for the herbarium. This house is situated next to the Botanical Garden in George and is run as a unit by the trustees. From those early days, the herbarium has grown to house over 10,000 specimens. For many years, Yvette was the volunteer curator of the herbarium with other local volunteers, helping her for 3 hours a week. We were joined by the botanist, Doreen Court, when she moved to George. Doreen was the author of The Succulent Flora of Southern Africa. Yvette, who did a marvelous job with great dedication for many years, was later followed by Priscilla Burgoyne, our first paid curator, who worked there for about 3 years. Since her departure, we have only volunteers continuing the work. Dr. Niels Jacobson is our scientist who deals with the IDs, assisted by 5 other members of the team, led by Ray Smith. One of the volunteers is only 17 years old. The duties include accepting new specimens, freezing, spraying with borax, followed by drying them in a drying cabinet. The volunteers then mount and send them on for labeling. This is followed by inputting data into Excel or Brahms. Originally, the data was put on Précis, then changed to Brahms, which we don't find very user-friendly. We received a lot of help from Hanley Snyman from Sanby, but it remains a difficult program and not for the faint-hearted. The Botanical Library, which is attached to the herbarium, had fallen into disarray during COVID and loss of staff, and it is now ably managed by Dr. Brendan Cole, who joined us recently. The extensive biodiversity of flora biomes in the Southern Cape make the potential for collection of many new species exciting. The Outram's crew group is currently the primary collector, together with Niels Jacobson. Most of the older specimens were collected by Yvette and Niels. We are where plant lovers come for help and advice. Members of the crew group make use of the collection when researching a genus or a species. There have been comments made that herbaria are no longer relevant and that everything will be online in the future. We feel that pressed specimens have much more to offer and so continue our work, but we do intend to upload our collection onto an online platform in the near future. Recently, a living collection was started by the curator of the Botanical Garden. These plants in the garden will have a provenance and will have accompanying herbarium specimens. Fundraising has not been successful, and we've only have a small budget from the Botanical Garden, which covers our basic needs. Our aim is to upgrade the facilities so that we can have closed cupboards and built-in work surfaces. From very humble beginnings, it has grown to a successful community herbarium. We are not affiliated to a university and are not a Sandby Botanical Garden, so the facility is somewhat underutilized. We are, however, proud to have a large collection of our local flora, and we strive to make it bigger and better. Thank you.
So that's a view from one of the smaller non-affiliated herbariums. And it's interesting that there are quite a few of these collections from around the country. And you would have heard briefly in Andre's presentation, and you'll hear again about the concept of orphan collections. So those would be herbaria that were started up at certain places and then have been consolidated into larger herbariums because it's just more efficient to run it that way. So we are going to move on now to KZN, to Pietermaritzburg. And I think also at the same time, it would be good to, if people on the call are close by to these herbariums, think of ways that you can also assist, even if it is from a moral point of view. But there is always a place for volunteers. Not a good day today.
Area situated in the school. Part is very important. We have to make sure that our temperature and humidity stays within certain limits. We want to prevent we can actually have crops survive.
Okay.
Rupert, I can
Yeah.
do
Now
the voice for
let me
that.
start
if you
that again.
want. Okay.
Let's see.
Good afternoon. The Buse Herbarium is situated in the School of Life Sciences at the University of KwaZulu-Natal on the Pietermaritzburg campus, one of the 5 campuses of UKZN. This herbarium has been going for as long as the university has been established, which is from about 1910. And we know this because we even have specimens that are labeled as the Natal University College Herbarium. And the Natal University College was the precursor to the University of Natal, which has now been incorporated into the University of KwaZulu-Natal. We are situated on the Life Sciences or Agriculture campus in Pietermaritzburg on the 2nd level of the John Buse B block. For those of you that have never seen a herbarium specimen, this is what we're looking at. This is an example of a dried pressed plant specimen, and you'll see that there's a label with lots of information about how and where and when the specimen was collected and by whom. And we also nowadays put barcodes on our specimens, which give them unique identifying features. As a teaching and research herbarium, we are a little bit different to some other herbaria, but we still fulfill the basic requirements of a herbarium. A museum for plants, or it's a library for plants, which is basically a collection of pressed plant specimens kept under archival conditions. And the archival part is very important. We have to make sure that our temperature and humidity stays within certain limits. We want to prevent especially insect outbreaks, because that will eat the dried specimens. And in this way, if we look after our specimens properly, we can actually have them survive for centuries. And some of the older herbaria in other parts of the world indeed have got specimens that are several hundred years old. A herbarium is basically a working laboratory for mostly plant taxonomy and plant systematic studies, which is the study of relationships between plants and how plants, which plants are closely related to others. It's a place where people go for plant identifications, for general plant studies. In our case, medicinal plant projects feature heavily, ecological studies as well. People come to the herbarium to match these specimens that they've collected for ecological projects. More and more climate change studies are being done on herbarium collections, chemical studies as well. So it's a very versatile collection, and possibly more than people realize. And to me, it actually forms the foundation stone of many, many botanical projects and botanical research in general. A view of our collection, so you can just see what environment we're working in. We're very lucky that our current building has housed us for about 40 years, and the collection was the herbarium collection space was designed by one of our previous curators, the well-known Prof. Olive Hilliard. We have lovely cupboards that seal very well, and that keeps many insect pests out. And in the herbarium, we basically try to get specimens from a state where they come into our oven with in plant presses being dried. Then once they're fully dried, they're processed, they're mounted onto card, they are photographed. We also digitize the collections. We transcribe the information into our BRAHMS, the Botanical Research and Herbarium Management System database. And then ultimately we file or accession our specimens into our cupboards in a way that we can find them again using an index card system. Our staffing, the permanent staff is actually only our curator, Prof. Benny Bitterbeer, who teaches and does research and curates as part of his job. Then myself, the principal technician or the collections manager, and then Ms. Prudence Magwaza, who's our herbarium technical assistant. But on the slide, we also have several people that have assisted us over the past few years. Ms. Tanya Mate was on a contract helping us to incorporate some what we call orphan collections. And Mr. Sifiso Ngati also worked on that project. Both of them sadly are now at WITS. Tanya is assisting them with an orphan collection, and Sifiso is, we're very pleased, has a permanent collection at their museum and herbarium. Simone Chetty currently is also assisting us on contract, finishing off some of that work. And here are two of our previous assistants. Mr. Dean Phillips did an internship here, and he did his master's here, and he's now the assistant curator at the Bolus Herbarium at UCT. And Sasha Ndursamy is also an ex-student that's assisted a lot. He's very much involved with crew and working with SAON. And I always like to say one of our best exports from our herbarium is actually staff. Not just staff, interns, students that have come through here. Because we are a smaller collection with fewer staff, anyone that comes in interns with us gets to learn a wide range of skills, and that sets them up for their future careers. The herbarium is a busy place, believe it or not. We occasionally do get time to go into the field and collect plants. This is our curator. He's an orchid expert, Benny, working on his specimens. And you'll see there's also little samples. We have spirit collections as well, where we collect the orchid flowers and alcohol. Simone checking out our index card system. Dean doing some photography as part of our digitizing. This is how we used to do it in the past. Prudence does all our mounting of specimens onto card, and she's also involved in some databasing and accessioning. And then Tanya, and also another one of our previous students and assistants, Nikisha Singh, who's now the curator at the Skuza Herbarium and Museum. So if you come to us, you'll go places, that's for sure. We also do some outreach. This is projects that we did through where we actually ran a plant pressing course in Durban as part of a BioBlitz initiative. Here we actually involved in the Happy Earth Festival at the local Sandby Botanical Garden. Here Benny is teaching the crew members from Ponderland how to digitize their collection, and that collection has subsequently come to us. Here we have some postgrads actually from agriculture, some plant and some animal science students that have come to work with us. They also collect vouchers for their research. In this photo, a new person is Ayanda, who was involved with Sandby, but she was based here for a while doing databasing. And this is our curator actually we were running a workshop for Sandby staff, Sandby grant staff from our local botanical garden. So while we do a lot of outreach, we mostly concentrate also on our own staff and our own students, since as part of the undergrad courses, we actually do some instruction, and then to the postgraduate students, we do instruction, particularly on collecting good vouchers. One of the things people don't realize is if you use a field guide, for example, Boone's Pooley's Trees book, every time you see a dot on the map in a decent field guide, that dot actually represents a herbarium specimen. So people love to make lists of plants and say, oh, I've done a plant list for that area. Your list of plants won't make it into a book. Well, it's very exceptional that your list of plants will make it into a book. If you want to have a dot on a map, it has to be verified by a specimen in a herbarium. So every time these kinds of field guides are actually produced, you are looking at herbarium data on those maps, and we mustn't forget that. The data that people collect here on their collector's label is really important data. And this data is also used in conservation. So here's a picture of the Hilton daisy, Gerbera aurantiaca, which is one of KZN's, sadly, flagship species because it's now limited in distribution. And it's through tracking herbarium data over time that we can actually inform research on vulnerable species. I've put this picture in because I do run sessions for the postgrads, especially the postgrads that work on natural products. There's a lot of focus on Indigenous knowledge systems, and on our campus in particular, there's a history of work on medicinal plants. Our collection actually has 2 historical medicinal plant collections, one collected by Father Mayer, who was based at the Marion Hill Monastery between 1890 and 1910. That collection came through the Natal Museum to us. And then there's another collection by Anthony Cunningham, who's one of the authors on the Zulu medicinal plant books that he also did in the 80s on medicinal plants. And we actually file that separately at the end of every genus. And then to add to it, any voucher specimens for plant research that involves natural products, chemicals, medicinal plants, and the like are also filed there. So it's very important that our postgrads know how to actually collect the voucher of the actual plant that they've worked on. And in some cases that can be challenging, especially when material is collected from multi-markets. So this is where the research aspect comes in and the teaching aspect, because some other herbaria might not have to grapple with these problems. Whereas it's lovely to say, well, we're not going to accept your projects simply whereas it's lovely to say, well, we're not going to accept your specimen if you don't have reproductive material on it, like flowers or fruit. We sometimes do have to actually accept vouchers from student projects simply because they have to be able to lodge their specimens somewhere. And depending on what they work on, it's not always possible to get those good specimens. So what we then do is we make a point of actually sending those specimens out on loan when experts borrow our specimens. And we do lend specimens all over the world to people that study plants. And we borrow specimens from all over the world as well. There's an international network of herbaria all over the world that work together. And through these collections, we actually exchange material for research. So we do send things on loan because sometimes the expert might be able to ID something even if we can't, and at least confirm the identification for the students. It also means that if this work has been published and that they've cited the specimens as being here, if ever the naming changes, if the relationships in those groups are found to be different to what we thought, they can always come back to the source specimen, the actual voucher that was lodged here, and look at it to see if it is still the same taxon that the work was done on, or whether we should be calling it something else. So from a research point of view, it's very important. The last thing I want to mention is that we've been heavily involved with the Natural Science Collection Facility for the last sort of 4 or 5 years. I've been on the working group for the collections management and conservation, which has actually resulted in a handbook being published, which is available as a PDF on the NSCF website. And that's why I'm going to leave the slide here for a while. Please take the time to visit the NSCF website. It's funded through the NRF, and for collections, both zoological and botanical and paleontological collections in our country, this grouping has been a game changer. It's managed to get a lot of us together to talk to each other. It's managed to get us to produce this book that sets the standard for collections, and it's a working document, so we will keep working on it. We were fortunate to get funding through the NSCF for more cupboards, which has allowed us to incorporate 3 smaller collections that needed new homes. And we also were funded with 2 staff members for 3 years to assist with that. So now the Killick Herbarium from KZN Wildlife in Pietermaritzburg is housed with us. The Pondoland Herbarium, which was based at Umtamvuna Nature Reserve, where this beautiful grassland slide was taken actually, is also now with us. So that's the Nicholson Abbott Herbarium. And then Our Westville campus had the Ward collection, which was mainly based on the work of the now late ecologist Roddy Ward. And that collection has a large wetland and mostly wetland collection, water plant collection focus, and then also a small focus on ecological samples that came from the old University of Natal Howard College herbarium. So this has increased our herbarium specimen holdings from something like 130 to 150,000, which we thought we had, to well over 200,000 specimens. We always have been, and now we certainly are the largest collection in KwaZulu-Natal. Our collection focuses on the Drakensberg and the KZN Midlands flora, but we do have representation from all over our province. And going further to the country, we have a fair amount, especially where grasses and sedges are concerned, we have a fair amount of material from tropical Africa and even extra Africa. So if you are a researcher, please do come and visit us or contact me. You see my details here if you ever need a loan. We also send images to people, especially during lockdown. That was very helpful. And then from the images, you can decide what you might need for your research. So we have a portion of our collection available already online for anyone to see. And I know that some people think that that comes with a risk. We're aware of the risks of making plant data available, but we're also aware that many people that do steal plants from the wild already know where they are. But we do hide localities for sensitive groups. And we hope soon, once we've managed to get more bandwidth on the university's server, to actually have a larger portion of our digitized collection available. And then hopefully once we've finished all our digitizing projects, we will actually know how many specimens we do have here. But certainly come visit us. We love to have visitors, and contact us if you want to know anything. If you're in KZN, I'm very happy to help anyone to learn how to press plants, to learn how to make vouchers and the like. That's part of our public outreach. So we aim to be approachable and we aim to be useful and helpful to the public. Thank you very much.
Thank you to Dr. Christina Curry-Potgieter. That was a really nice overview, and it shows the important fact that the herbarium is not just a collection and curation for specimens, but as she was saying, a conveyor belt for people working in the sector. So training and moving on. And it just when people ask, what is the use of a collection like this? That for me, as a people building organism, is an important function of the herbarium. I think we've figured the sound thing out now. So I'm gonna have another go at Andre's presentation, and please give me the thing is it's a PowerPoint, and I have to play the sound from each one. So I'm gonna ask my colleagues on WhatsApp to just send me a message to see if it works. So let's go and check it out.
Good day everyone, my name is Anri Maretianus and I am the technician for science collections at the Compton Herbarium at Kirstenbosch Research Center. Today I would just like to explain to you what the herbarium is, what we do on a day-to-day basis. The herbarium was founded in 1937 by Professor Robert Compton. The South African Museum, which is also the oldest museum in Africa, was incorporated into the herbarium in 1957 and then the Stellenbosch Herbarium in 1996, thus making Compton Herbarium the second largest herbarium in South Africa. Compton Herbarium's collection consists mainly of dried plant specimens, but we also have DNA material, wood samples, leaf material. We have a collection of seeds, as well as a collection of maps and historical books and botanical art. So at the herbarium, we do a whole lot of things. We provide a lot of services. We do processing of incoming specimens, which could be loan or gift material or donations. It could also be an orphan collection, and we do all the curatorial processes behind it, all the administration. We also provide identification services to individuals and many other institutions, such as universities or nurseries, etc. We also charge a handling fee for every specimen being identified. We do pressing, drying and mounting of collected specimens before we incorporate it into the herbarium. We've recently started digitizing or imaging our collection. We are currently above 80% completed, and this is just using a UV light box where we put a specimen in and capturing the image of it, and this is for us to create basically a virtual library or museum. So come a natural disaster or anything, then we will have all the electronic copies of every specimen in our herbarium. We do identification and description of new species. We also produce taxonomic revisions, and we do provide working opportunities or working space for researchers and visitors as well as students. We also do loan agreements with other herbaria, both national and international. We also provide outreach programs throughout the year for learners and students and visitors, but this is by appointment only. Here's just some pictures of the entrance to the herbarium, and as you can see in the pictures, these are all metal cabinets. So a couple of years ago, or back in the day, the herbaria used to make use of wooden cabinets, but it's since been replaced with the metal ones instead, which obviously provides better protection and preservation of the specimens. And then on the pictures on the right side of the slide, you can see all the yellow folders with the specimens, which are all filed using a numerical or the APG2 system, which we can use to easily track our specimens. Bear in mind we have about approximately 1 million specimens in our collection, so we definitely need some system to track each and every specimen. And these are all filed. You can see all the pigeonholes with the specimens. They're not clustered or compact. We try to space out our specimens to prevent any damage and avoid any damage to the specimens. So the top images, you can see some wooden cabinets, and there's only a few wooden cabinets left with specimens which is currently stored in the South African Museum, but these will soon be replaced with metal ones as well. And then on the bottom images of the slide, you can see the wooden samples, or the wood samples rather, of our collection, and these are all barcoded and also have their labels with information on. Our leaf material collection is stored in our freezers, which is stored in the preparation room. Our DNA samples are stored in the freezer, which is a minus 80 degrees Celsius freezer, and this is stored in the anatomy lab. The seeds collection are stored in these metal cabinets with the drawers, and on the right side of the slide you'll see a large metal cabinet. This is where we store our maps collections. When it comes to collecting, pressing and drying specimens, it may seem like a daunting task, but if you know what to do, it could be a breeze. It's very important to know what needs to be collected and how many. Remember, different plant species have different identifying or diagnostic characters. I'll just give you an example. Succulents, you need the fruits to identify, so it's quite pointless collecting a succulent plant and only collecting the leaves of it or something that's in flower. You will definitely need the fruits to be able to identify your species. For iridaceae, for instance, you need the bulbs to identify, so it's pointless collecting an irid that only have flowers and then you don't have the bulb. So it's quite important to know beforehand when you go to the field and you are collecting, to know what you need to collect and also what parts of the plant you need to collect. It's important to try and collect plants in flower. Try to get all parts, such as the leaves and the flowers, the fruits, the bulb and the root, etc. Also note that drying times of plants vary. For instance, succulents take longer to dry and requires constant checking. This means checking up on the specimen basically on a day-to-day basis, and also changing the flimsies or the newspaper of in which the specimen is pressed, changing it every day to prevent mold from forming. And this is just placing a new dry flimsy around your specimen every day, and this will prevent any mold or moisture just sitting there and building up. In the event of mold forming and you've detected it quite early, you can still treat it with 70% ethanol. You just use a little brush, dip it in the ethanol and just gently brush over the specimen, and hopefully this will kill off all of the mold. In this slide, you can see in the top right an image of a plant press, and this is basically just two wooden blocks or structures, and between the two wooden blocks you'd find corrugated cardboard and absorbent paper and unprinted newspaper or newsprint flimsies. This is just, the wooden blocks provide structure for the specimens that you would then sandwich in between. So it's basically repeating the process of placing your fresh specimen inside of a flimsy, then sandwich it between two absorbent papers, which is then sandwiched between two cardboards, and that's how you repeat the process for every specimen. Depending on the amount of plants that you collect, or also the thickness of the plants, and also the length of the press straps, you can add multiple specimens to a plant press. Just make sure that you add enough material in between so that moisture doesn't build up. Remember to add a collecting label to every specimen, and this is just a label that contains all of the relevant information of your collection, such as the collector name and the collector's information, the date of collection. The locality information is quite important. If you can have or get GPS coordinates, that's always helpful. And then just basic habitat information and soil types, etc. Collect enough material so that it's able to fit or fill an A3 mounting board should your specimen be identified and incorporated into the herbarium. The bottom two images on the slide shows the dryer with the pressed plant material. So this dryer is stored in our preparation room, and it can fit quite a few presses, as you can see, and this has a constant temperature which provides the airflow, and this ensures that the specimens stay dry and dry out quite well. Mounting of specimens may seem like a very time-consuming task, but it's also very therapeutic, and it plays quite an important role. Mounting prevents damage and loss of material. If we had just kept all our collected and dried specimens in their newsprint flimsies and they stay there for years, it would be so easy for plant material to, fall out and just get lost in the stacks of all the specimens. So mounting of specimens prevents that, because you are mounting the specimen on a mounting board by using these gum strips and ensuring that the parts of the specimen stay in place and intact. It also makes it easier to handle because they are mounted on a board which is quite sturdy. The other aim of mounting is that it ensures better objective of the parts of the plant. So you can see all the details of the flower and especially the label information. So it's quite important that we mount all of our specimens before we incorporate it into the herbarium. Most of our mounting are done by volunteers. We do have students and interns doing mounting as well. Some of the staff members also performs mounting. Mounting does pile up quite a bit because it's such a time-consuming task and it takes quite some time to mount a specimen, but we try and do our best. So we're always in need of volunteers. So if you want to come and volunteer at the Compton Herbarium, please contact us. So once our specimens are mounted, they are ready to be incorporated into the herbarium. But before we do so, they have to be decontaminated first. And we use these two chest freezers in the bottom left corner of the slide. We place our specimens in plastic bags and seal them up, or in boxes. And we place it in the freezer for at least 72 hours, and this just ensures that it kills off all the little pests and insects and bugs. We also do the decontaminating of all other incoming specimens, such as loans and gifts and donations. So we definitely always make sure that we place the specimens in the freezer before they go to the first floor, which is the herbarium, before they get incorporated into the cupboards in the herbarium. So what makes a good specimen? It's important to know that your specimen needs to be of good quality and that it is the correct size. It's pointless collecting something that is entirely too small. That way you won't be able to see all the required parts of the plant. So it's best to do some research before you collect. It's important to collect all the required parts of the plant, relating back to one of the previous slides. So if you are collecting succulents, make sure that you are collecting the fruits. If you are collecting anything in the Iridaceae related family, make sure that you are collecting the bulbs of the plant. It's good to always try and collect something in flower. Make sure that your specimen is properly pressed and dried, and also ensuring that no mold has formed on your specimen. It's always good to add additional fruits and flowers, and this is just for dissecting purposes and also for rehydration. So a lot of times the taxonomist or the scientist who is performing the identification of the specimen may need some extra material such as flowers, and they may rehydrate it for being able to see different parts, the anthers and the ovaries of the flower or the plant. So that way they won't be able to remove all the flowers off the plant specimen, and having additional parts of it just ensures that they can have extra material to work with. Lastly, it's important to know that your label has all the proper and correct information. So it just makes the identification process a bit easier if the researcher knows where the plant was found and the soil type, the habitat, also what time it was in flower, etc. So it's important to note down all the proper label information. And that's about it. If you are a student or a researcher or just a visitor who would like to visit the herbarium and get to know more of Compton Herbarium, please feel free to contact us. You may contact the herbarium coordinator, Mrs. Kuliwe Dubula, and there's her email details are in the slide. You can also email compton.herbarium@sanbi.org.za. And yeah, this is just for anyone who's interested in what we do, especially if you are a student who would like to know more about the herbarium and everything it is that we do, maybe different career paths for school learners. You're more than welcome to contact us. Thank you very much.
Thank you, Henri, for giving us the behind the scenes view from Compton. Very much appreciate seeing those drying cabinets and so forth that I've been there since I've been knocking around the building. And that's another good thing, is that if things are well made, then they stick around. Okay, so we've finished with the presentations. I'm just checking to see, are there any questions for the presenters that you have? I'm going to ask Henri, Christina, and Jenny to put your cameras on, and so that we can have a quick discussion. I've got an immediate question for Henri, actually. Just
Sorry, guys. I'm having load shedding. I just
okay.
put on my emergency light now, so sorry for the
Ah,
shadows.
I think it might actually be better if you switch off your video, because then your sound will be better. But I just wanted to check, explain the rehydration of a specimen.
Okay, so I've actually just heard this the other day when I was doing some research for this presentation. So it turns out that Dr. John Manning, who works on iridaceae, so he sometimes need additional flowering material, and what they do is they kind of boil it down. They didn't go into too much detail, but that's basically to just rehydrate the specimen so they are able to look inside the flower and are able to measure the length of the anthers and the stamens, etc. I found it quite fascinating because I actually didn't know that that was being performed actually.
Yeah. No, I mean, you assume that when things are dry, then they stay dry forever. So that's interesting.
Yeah.
Okay. Jenny, or Christina, do you have questions for each other before we put it open to the audience? Audience, you're welcome to put questions in, and Patience, you can stick your question in the Q&A. It's fine.
May I ask a question of Christina?
Go for it.
I'm very interested on your index system. Can you just give me a brief idea of how that works?
Okay, so the numbering that we use is a numbering system that many herbaria have used. It's based on a book by De La Torre and Harms. And then we use that, but our index system is alphabetical. So we have a card system where you can find the genus by alphabetically, and then within the genus, the species cards are arranged alphabetically, and then those have the numbers on them that then take you to the appropriate cupboard.
Okay.
But more and more, since molecular studies have been showing that some of how we previously thought different families even were related to each other, molecular studies have turned a lot of that on its head. Some herbaria are simply choosing to go alphabetical by family or genus. And others have taken on the angiosperm phylogeny groups classification systems. But the problem is there was APG1, APG2, we're on APG3. So, are you going to rearrange your collection every time? So I think whatever system you have, just have a system where you can find the specimens and stick to it. And as long as you can find it, I think that's the most important thing. And people that are frequent users will learn where to find the genera, the families that they're interested in.
I like the idea of your card system. I think we must make some changes. The other issue is when genuses are changed, like I believe Ortholobium or now Soralia. So now do we go back and do we change the whole lot? I mean, how do you ever keep up, and where do you always get the latest information on what's been changed?
Well, if I can answer that, Rupert, we are very lucky in South Africa that SANBI does a lot of that for us, and more they are making it available. SANBI actually has a person, Ronald Klopper, who is employed to keep up the list for South African plants. So whenever there are changes, it's incorporated. Now, sometimes people don't agree with some of the published changes. So there's actually a panel made up of 3 SANBI people, 3 non-SANBI taxonomists, and the non-SANBI people are actually voted on. And then Ronel has the last vote to decide if there is a very controversial paper that comes out, the majority might choose not to adopt it for the national list. But if you keep in touch with Ronel, she can send you updates of that list, and that is quite useful. I think we must realize we're always going to be out of date. So, and we've noticed that when we've incorporated these orphan collections now, sometimes they were more up to date than we were. And once again, as long as you have a system where you can find your specimens, that's fine. If you're using an old name, it's not wrong, as long as people can. So what we do in our card system is if names do get updated, we put the old names in square brackets and we put the new name. And then if the numbering is changed, we change it, but I tend to change it in pencil now. Because I know
that's probably a good idea.
And then redo it. So some changes are just they take an enormous amount of time. So you just have to decide where you're going to spend your time. And as long as people can look up the new names and the old names, it's actually not a problem.
Well, that's the thing, is to have access to that information. But I'll talk to Rupert about that at another time. We don't quite know where to get that information.
Well,
thank you very much.
Talk to Andri. So I've actually got a question for Andri now, out of the chat, from Patricia Chatukuta. What are the challenges you have experienced in digitizing specimens, and how have you overcome those challenges? So Andri, as you are.
I'm here. Cool. So currently with digitizing of specimens, obviously load shedding. So that just, throws a whole rock in the middle of the road. But other than that, we've been quite on track with all the goals we've set for the year, and we've actually managed to do quite some digitization. We make use, I didn't include any pictures in my presentation, but I think Christina did, of the UV box that we are using. And the only issue that we're having is that, it's quite slow. If you have about 1 million specimens in the herbarium, is you have to do it one by one. But we are in the process, or so I've heard, that the new machine has arrived in South Africa, and it's basically like a conveyor belt where you can load multiple specimens on, and it does the imaging for you. So that's basically it. I can't really think of any challenges that we've been experiencing other than, the load shedding and us not being able to image as fast as we'd like. But yeah, the collection's almost completed. And then I think that's just something brilliant. I think we'll move on to the South African Museum next after our collection is digitized and imaged.
So that then leads us on to a second question that you might have seen in the chat from Tony, because the question is it possible to move some collections to the top of the digitization queue? Because it would be good to get Purcell's collection from Bergfleet digitized. So does he need to pay a bribery fee, or how does the process work in terms of getting things into
Tony,
the line?
you can definitely contact me. We can work out a bribing fee over there. Actually, so I will need to check up on the Purcell collection. Like, I'm not sure what the person collected or how many the person has collected. I'm not very familiar with that. But the thing is, so when we do digitization or imaging, we don't do it per collector. So we are actually just going row by row at the moment, which basically is family by family. So we basically started with Scrophulariaceae, and we are currently doing Iridaceae at the moment. So it is possible for him to probably get an extract of that person's collection, like the virtual copy of it. But then we'll just need to make sure that they are all, most of them would be databased and in Brahms. So if we can just pull an extract of that person's collection, we should be able to get hold of the images, because when we image the specimen, we don't save the name as the collector, etc. So as you noticed on our specimens, we have barcodes and they all have a unique ID. So when we capture the image of a specimen, we save the name of that image as the barcode number. So then it would be easy for us to track the name if we can just match it up on the database, and then we can just do an extract based on the barcode number instead. Yeah, I hope that helps.
Yeah, cool. Thank you.
All right.
So Petra Brodel in the chat reminds us that APG4 exists. So that's one of the the classification system, and essentially she also said that the name changes are based on publication. So once you got the publication, that gives you the indication for the name. That's more for people who are unaware of that process. Okay. I'm just checking now. Oh, okay. There is a solution. We've already solved one thing in this webinar. Ronald is the oracle, and the Ronald's email address has been made available to people who need it. So I think that's great from a service provision point of view. Jenny, I hope you're happy.
Yes, I'm very happy. Thank you so much. Thank
Christina,
you.
go for it.
Can I just say to people, don't neglect the Sanby website. From when I started working here over 20 years ago, when Sanby held on to all the information and no, you couldn't get anything, it's completely changed. They make everything available. They give us Brahms updates. They give us updates of all sorts of things. You can get so much from them. If you are using Brahms for databasing, they give you, you can get files with localities and all sorts. So there's so much free stuff available on the Sanby website. It's scary. So people must keep looking there. And at the same time, on the NECF website as well, there's webinars, there's videos of all the collections, but that includes zoological collections as well, though. But there's so much information available. People just have to go and look for it. Sometimes you just don't know it's available. But if you, and I do wish Sanby would organize some of their publications a bit better on their website, but they are there. So just look. We're very lucky. Things have changed a lot.
Yeah, no, I think +1 from my side. The Sanby website has got such an amazing amount of cool stuff on it, including publications and the new books and so forth. So you definitely do not need to go much further. That's the spot where we are getting our taxpayers' worth. And I just wanted to thank all the speakers. I don't see any last questions, but thank you, Christina and Jenny and Anri, for being so generous with your time and data and dealing with the various load shedding issues. And I want to remind people that the next webinar is on the 23rd of February, and it's basically a sequel to this one. So we'll go from the buildings, housing collections to taxonomy. And so everyone who's been frustrated about changes such as the Ortholobium Soralia thing, log in and let them have it. We're going to have a couple of taxonomists online. And maybe then we'll be a little bit more sympathetic to the things that they've got to deal with. So thank you for joining us. Thank you for sticking through the technical glitches, and we hope that everyone has a great month going forward. Thank you so much. Take care.
Thank you.